Hello, everybody. Now we are going to start finding overlaps in our ChIPseq data. So in this class, we're going to use genomic ranges and learn the basics and then apply it to our pull2 ChIPseq data we generated off the Nextflow pipeline. So this would be really a common first step for anybody we analyzed 4 replicates, and we'd want to say how many peaks were in each replicate and how many overlapped, etc. And then we're going to move from finding those overlaps within the peaks of our ChIPseq data to genomic features, such as a promoter or a gene body or your favorite transposon, whatever you want. We will go into how to use genomic ranges on genomic annotations of all kinds. So in short, the first thing we're going to do is learn how you would take your peak file and compare it to another peak file and find the overlaps between 2 samples. And so we had a tutorial on Iranges. The link is in the description, but GRanges, genomic ranges, or I'll say GRanges, is based on Iranges. And so if you want to get a tutorial on that, you can look back there. So let's get started. We are in classes, class 2023. Our basics, and now we are on 04 genomic ranges here. And so I'm going to run this in our studio, but that is the same code that's on the GitHub. Okay, let's get started. So Iranges, we went over, and it tells you the start and stop of a given feature, such as a peak. But in the genome, there's a problem with the multiple chromosomes. So we have a start and stop that could be the same values on chromosome 21 and 22, so we need to keep track of that and add an extra layer to Iranges. And that's what Genomic Ranges package has done. It's pretty much what I use exclusively. I don't know if that's outdated, but it does pretty much everything we need. Okay. So as far as analyzing peaks, etc. Okay, so genomic ranges is like we set up before with Iranges. Here where it's saying Iranges start, and we went over this in the last class. Okay, so now what we're doing is we're just sort of wrapping that Irange function that's highlighted here, and we're telling it some new things, like what the chromosome name is. That's the most important thing. And so we have this extra variable seq names, which is super handy. We'll see more later. So if we look at this from left to right, we're saying call up the GRange function. So we're going to make a range of starts and stops that are associated with the chromosome. It also can track the strand if you're on the plus minus strand, or you don't know the strand, the star, which is the case for us with ChIP data. And then you need to set the start and stops of the values for that chromosome's attached and value. Okay, so let's make that here. So we now have our first GRange, and you can see it's kind of complicated. You'll get more used to reading it in here as we go along, but let's just first start with seeing what it looks like here. So we can see that we have chromosome X here, we have Iranges, and the run length encoded. We'll go over run length encoded later with the metaplots class. It's cool, but again, you always want to, last time we had just integer, now we have a more complex sort of data form, so it's good to keep track of that. The start and stop, and this one's on the plus, minus, and star strand is the order we made it. And what we're going to use is GRange to read in our peak files. I'm just sort of showing you what it's thinking about in a basic example. And so if we look at the seq info, which is pretty handy, these are all the variables that can be attached with the chromosome, like the chromosome name, the number of its length, is it a circular chromosome, and what genome are you working in. The genome is pretty important because a lot of this code could look similar for a mouse genome or a human genome. We're using exclusively the human genome for this class. So we can look at what this name is and set a value for it. So let's just set an arbitrary value for the X chromosome. And now when we look at that, it is now placed in there. And this is really good to use these seek levels when you read in data, just to see what they have associated with it, and also seek names. And we'll do that in just a minute. Okay, so let's add another chromosome here. We can make X and Y and take a look at that. And we now have 2 chromosomes, and you can go on and on. But genomic ranges will just have this header for, or meta bin for, a chromosome name, no matter what species you're in. Okay, and then we can add the genome if we want here with HG38. It's the one we're working with. And now we can see we have a genome value down here. Okay, so let's actually get our data now and see what GRanges does with it, now that we know how it sort of formatted, or its syntax. I'm doing this so we don't overwrite folders. I'm going to put this back to your folder. And you should go to your folder wherever you cloned this class on your computer. That's where that is. And then this is where we ran the Nextflow pipeline on our ChIPseq data. This should probably be the same for everybody if you have followed along the same way. So what I'm doing here is making 2 objects that we're going to paste together using file path. I'm simply doing this so we don't end up going into each other's directories, so that this is specified by you. There's issues with that in the previous classes. So I apologize for this, but it's good to know in base R, file path is going to take a character string and go to that place. And it's like paste, where you can add a bunch of features to get to the actual file path. And so what we're doing here is we're making base path, and file path is going to merge those 2. And then what I'm looking for is this peak file that you also have here. Classes 03 Nextflow, oops, not that one. The Pull2 NF core run that we all did together. All the data is online. Okay, in results, BWA, merge library. We'll get used to this later, but I'm just showing you where that path is actually going. Broad peak, and then here's that file, pull_1 peaks.broadpeak, peaks.broadpeak here. It's important because Nextflow or NF core gave us a bunch of different file formats that are useful, so we just want to make sure we grab the right one. And so what we've basically done is just set a file path to go grab those. Okay, and I'm going to call it peaks1 and peaks2 as the object name for the environment. And now we can see them populating in the environment. And it's good to start looking in here and seeing what is all in here. We can see the chromosome names, some weird ones, which we'll get rid of in a second. And names of peaks, score, signal, p-value, q-value. We've seen this before in the class where we read in a peak file. So that's good. It's all in there. You can we'll start looking at it in different ways, but one thing you can see right away is that peaks1 or replicate 1 had 33,582 peaks, and this one had 43,000. So there's a bit of information right away. We do have 4 files. I just grabbed 2, so you can copy and paste and just switch the 1, 2, and add a 3 and 4. Okay, so and then if we, just for reference, if we had read this in with a TSV like we did before, what we're going to see is this peak file. And remember, we had to do the row name, column names equals false, because what it did is it added our data to is a column. These black regions are not actually in the data frame, they're the column names. So that is all basically being stored in peaks here, right here in the list data. Is going to have the same sort of values as this data frame. Okay, and it gets confusing for a while, but then you realize how handy all that information actually is. Okay, so we have our 2 peak files. We just compared it, was reading it in as a TSV. We would normally want to do change the call names equals false here, so we don't add that there. We've been over that. Okay, so find overlaps can just take is a genomic ranges functions, and we can just find the overlaps between the entire peak sets of 33,000 and 43,000. And so let's look at that real quick. And we've done our first overlaps. And what's interesting to note is this warning here is not a big deal. It's saying there's a bunch of weird things that don't look like chromosomes. That's true. There's parts of the genome that are in contigs where they don't know what chromosome it belongs to. And so we'll clean those up whenever we read in peak files. Okay, so we can look at the length of this. We kind of already know this from the environment here, but length and width are very common things to check on a genomic ranges. So this is just going to tell you how many peaks there are. And if we look at the overlaps, there's 28,000. So that's kind of a fun start here. We can see that replicate 1 had 33, 43, and we got 28,000 peaks. So a good majority of them sort of overlapped. All right, so that's good news. Maybe our replicates are looking good. And we can also do the summary, which is sometimes even more helpful. In this case, it's not very helpful of the overlap. And it's just telling us that it's a GRange object with 5 metadata columns, and the overlaps had none because it's just the overlap regions. Okay, so this is great, and it makes sense because we have less overlaps than we do peaks. But imagine the case where you have more overlaps than peaks in a given file. If you think about that for a second, it's unusual, but it happens all the time. It did not happen to us here. So I want to do a quick little thought exercise using a data set that's actually from POL-R2A versus POL-R2B, and see how we would troubleshoot what's going on there. And that'll get us to learn a little bit more about genomic ranges, specifically this @to and @from. But to summarize, you would basically just read in your peak files and do find overlaps between your peak files, and then take a look at them and find the overlap number. And that would be a good start to your overlap results. So let's read these in. If you're in class, if you're not, you can download them here with the wget real quick in the same, or just keep track of the directories. I'm going to call these crazy peaks 1 and crazy peaks 2. We're doing the same thing. We just read in the exact same broad peak type files. And we can see here that we have the seq info with the chromosome names. We have the ranges here where the start and stops and widths of all of them are. They're in kind of a vector format, so it's not as intuitive at this point. So we'll get more and more familiar with this. Okay, so we've just read these in, and we're gonna do find overlaps. So same process that you would always do is just quickly find the overlaps. And we did this same kind of exercise above, where we do summary. We got 10,000 peaks in our first one. I'm just showing you the summary, and length can tell you the same thing. Length here, 19,000. So basically we have 10,000. Let's do length just to show you it gets the same number. We get 10,000 in one sample, 19,000 in another sample, but we get 11,000 overlaps between these two samples. How can that be? Well, there's a couple of scenarios. I've outlined them here, and we can go over that. And basically, it could be that all the peaks overlapped from sample 1 in sample 2, but a few had more than one overlap. That'd be amazing. But it also could be that very few peaks from sample 1 had many overlaps with a region in sample 2. So we got to those are very different outcomes. And so we want to really get in and think about this. So let's look at the width function. We use length to get the number of peaks. And if we look at the summary of widths here, what we're seeing is how wide the summary of all the peak widths. And let's do the same thing for peaks 2. And what we can see here is that in sample 1, the peaks are twice as big. They're twice as long on average. So if it's a longer peak in one sample and maybe two peaks right next to each other in a second sample, that bigger peak is going to count 2 times that it had 2 different overlaps with peak 1 and peak 2 in the other sample. And that's why it's really important to think about which way you did the overlaps, which we talked about before, which one's the subject, which one's the query. And we're going to go into that a little bit more with @to and @from. And it's really important. I know it's going to seem esoteric, but you can make really big mistakes if you're not paying attention to which way you're comparing your data. Okay, so @from is the subject side. If you want to think about the perspective of the first file you did in find overlaps, and @to is the perspective of the other sample that was overlapped with. And this is just an example here that if you had a big peak in sample 1 and 2 peaks that don't overlap, these are the stars are for the peaks in sample 2. And so 3 peaks overlap here. And so what we can see is from the sample 1 perspective, it had one overlap, window 1 had one overlap there. And @to would say, oh, I had 3 overlaps there. So we want to be careful, and we'll take a look at this now here. So if we look at this crazy overlap of peaks 1 and peaks 2, this isn't very helpful, but this is telling you how many times each peak overlapped in the index. And so if we do table, we get a more better way of looking at this. So you can table the table here. And in this case, it's super helpful because these numbers right here are actually how we get to 11,000 instead of the first sample only had 10,000 peaks. So how do we get 11,000 overlaps? Well, we can see that 6,000 only had a unique overlap. So that's a very different result where we didn't know if it was all 10,000 or 200 or whatever. So it's good to know now that 6,000 peaks had a single overlap, and you might want to take that into consideration for your analyses. And then also 918 had 2 overlaps. So if you do 2 times 918 plus 6,388, I think I have the math done down here. If you add these up and multiply them through and add them up, you get this number of 11,042, which was the peak overlaps. So it's important to note that the overlap number is from a perspective of the subject into the query, and it's good to remember which way you're looking at this here. So if we look at @to, we can see that in this data set's perspective, most of its peaks only had one overlap, 10,996, and 2 had 23 had 2 overlaps. So that's way less overlaps. And this is probably due to the wide peaks in sample 1, had many more overlaps with sample 2. Hopefully, if we added another replicate 3 and 4, we would start to clean that up. But I think it's good to see how the overlap number can seem much bigger if the peaks are bigger in one thing and not the other. And you might be looking for more of unique overlaps. So as another solution, instead of looking at @to and @from, we could have simply counted the overlaps to get this answer. So if we use now the example of count overlaps, before we used find overlaps, now we're going to look at count overlaps, and they're all really handy genomic ranges things. So this is where we're going to count them, but we're not going to know the positions of where the overlaps were. So we won't have the @to and @from information. I prefer find overlaps whenever I can, or subset by overlaps is another example we'll use later. But with count overlaps, it's a quick way of seeing what happened here. So if we look at count overlaps and call this crazy OVF2, we see that it's actually a value. It doesn't come up as a GRanges in our environment here. And it's the 10,000 peaks in sample 1 that we had as our subject or query subject into the query. And so we can see right away that the first entry had 1 overlap, the next one had 2 overlaps, and the 5th one had no overlaps. And it's just going from peak 1 all the way through the list. and tells you the number of them. So if you wanted to get that unique number, we could just sum this vector. That's using the sum function here, and we'd see the 11,042 overlaps, which is this math down here, if you want to follow that along. Okay, so if we did table again, we would see the same thing, that 6,000 had one overlap, and then you would also know that 2,242 had no overlaps, and then how many had multiple overlaps. So that's important information inside the not just finding overlaps, saying, okay, I have this many overlaps. There's a little more to it, and it's good to know which way you're looking and which samples which. And so for this exercise here, I want you to try and take all 4 of the POL2 replicates and do find overlaps and intersect all 4 of them and see how many, report back sort of how many peaks were in each file. You can use length or look in the environment, and then do the find overlaps, and then do another find overlaps as a hint in another one, and then see how many peaks you get that overlap in all of your samples. And with that, we will then move on to see where these overlapping peaks are in the human genome with respect to promoters or gene bodies or enhancers or whatever you're interested in. That is where we're going next. Okay, until then, be well and thank you.